Thank you all for joining us this afternoon. I'm Suzanne Godby-Inglesby, Associate Director of IU's Institute for Advanced Study. And today we are pleased to host Dr. Paul Rothrock to discuss Plant Photographic Scavenger Hunt, Enhancing the IU Herbarium Digital Archive. Before we get started with our program, the IU Institute for Advanced Study recognizes that Indiana University is built on the ancestral homelands and resources of the Miami, Delaware, Potawatomi, and Shawnee people. We acknowledge and honor these indigenous communities, both past and present. Our Resilience and Memory in Archives, Libraries, and Museums series will continue with a February 11th presentation by Dr. Gwenera Isaacs of the Smithsonian Institution's Anthropology Department. We'll be discussing plaster face masks made in the 19th and 20th century. To register for that webinar, please click the link that we will add momentarily in the comments. The format of today's presentation will be a presentation by Dr. Rothrock, followed by a question and answer period. And those of you who are here with us will have the opportunity to put questions and comments in the question and answer section of our meeting here, which should appear at the bottom. And I will share those with our presenter at the end. This event will be recorded and available for later access on our website and YouTube channel. I would like to acknowledge the co-sponsors of this year's Resilience and Memory in Archives, Libraries, and Museums series, the IU Themester, and we thank the IU Herbarium for being a partner for this particular project and presentation today. Dr. Paul Rothrock is Associate Curator Emeritus of the IU Herbarium. Founded in 1885, the herbarium houses over 161,000 specimens. These include physical collections that may be studied in person and a growing database of digital records and images. The digital archive is a particularly useful tool for citizen scientists in the state of Indiana and beyond. In addition to his other research, such as his recently published second volume of a richly illustrated guide to the sedges of Indiana and surrounding states, Dr. Rothrock has been a key player in expanding the herbarium's digital holdings. Today, he will discuss a crowdsourcing project launched in 2021 to expand this archival data. Dr. Rothrock, welcome. Thank you for being with us today.
Thank you, Suzanne, for that lovely introduction, and especially for the opportunity to share our plant photographic scavenger hunt with you in the audience. Before I get this to advance. Here we go. Before I get into the core of the presentation on the scavenger hunt itself, I'd like to provide some context. First of all, what is an herbarium, and a brief history of the IU Herbarium in particular. The effort that we took over a 5-year period in creating our digital archive to stand alongside of our physical archive. And in particular, I want to spend a little bit of time showing you the web platform that houses our digital archive. It's called Symbiota. And then we'll get to the why and the how of the scavenger hunt and the outcomes of that project. First of all, what is an herbarium? In this image, we have a typical herbarium specimen, and you can see there is a pressed, dried plant, a physical specimen that has been mounted on document quality paper that measures 11.5 by 16.5 inches. And in the lower right-hand corner is a label that contains the identification of the plant, the date when it was collected, the habitat of the plant, its physical locality, and who did the collecting. In this case, it was Charles C. Dean, who collected this plant, and its number 22,536. He was a very busy man. He did collect over 60-some thousand specimens before he passed away in 1953. As well as the label that he provided, notice that above it is an updated slip that I've added, which changes the name to Splenium rhizophyllum, indicating that there's been a change in the taxonomy that is applied to this particular species. Dean correctly identified it, but we have modified how we've classified this particular specimen. So this is one of 161,000 specimens that we have in the herbarium, and they are arranged in folders, and they're ranges of a huge filing system that can be easily searched and retrieved to see what the specimens are like. So an herbarium is in essence a plant library, but it's unusual in that a library would have copies of books that are all the same. But we have, each specimen is unique. It's each a genetically distinct individual. And so it's showing something about the variation of that particular species of plant. We can study the fine detail of these plant specimens far beyond what you can see even in this high resolution image, even though it's a very fine image indeed. In recent years, we've also been able to do modern science on these specimens, where many of them can be used as a source of extractable DNA. So you can do molecular work to see how they compare with other organisms. Let me turn now to some of the history of the IU herbarium in particular. As Suzanne indicated in introduction, our herbarium was founded in 1885, and she indicated it had 161,000 specimens. I say 159, but that's only the vascular plant specimen records. So we do have some specimens that are algae, fungi, mosses, and those add to that total. Of the 159,000 vascular plant specimens, though, about 1/3 of these came from the work of Charles and Stella Dean, as especially Charles was working on the flora of Indiana back in the early part of the 20th century. So as he was wrapping up that work in the late 1930s, he started to, he sold the herbarium and started to come to the university in allotments starting in the late 1930s and on into the early 1940s. Notice though that when he sold his herbarium to IU, our local collections were about 44,000 specimens. So Dean's edition more than doubled the size of the Indiana University herbarium at that point in time. But in addition to the significant holdings that we have from Charles Dean and Stella, there are other voucher materials that have come from IU faculty over the years. And these are also extremely important holdings in our repository. So for example, we have about 1,900 specimens that are vouchering the work by Ralph Cleland on the genus Enothera. This was groundbreaking research that led to his being elected to the National Academy of Science in 1942. We also have about 7,000 specimens that came from Charles Heiser and his students. Now, Heiser did a lot of work that dealt with economic plants. So, for example, the genus Helianthus is sunflower, capsicums are hot peppers, solanum would include tomatillia, and Laganaria would be gourds. And Heiser's work with these kinds of economic plants led to his also being elected to the National Academy of Science. And then finally, we have an extensive fern collection from the work of Gerald Gaston, who retired about 8 years ago. And also we have, we're adding now collections of my own that deal with the family of sedges, as well as those of the director of the herbarium, whose work deals with giant lobelia and other members of the lobelia family. So it is a very significant collection, both in terms of the flora of Indiana, but also globally in terms of vouchering the research by IU faculty and others. The IU herbarium was housed in Jordan Hall until the late 1990s. At that point, because of the high real estate value in the central campus, the archive was moved to the east edge of campus and is now housed in the Smith Research Center. The move, however, has some implications. One is that it led over time to a rather low level of curatorial support and reduced usage because of that remote location on campus. And as is true of herbaria in general, up until about 20 years ago, these were not user-friendly to the general public or the citizen science. What was needed was to bring the herbarium into the 21st century and to basically build a modern digital archive that would stand alongside of the physical archive. And so starting in 2014, we began to develop the information system that I want to tell you a little bit about this afternoon. So our digital archive is uses the web platform referred to as Symbiota, which was a platform funded by the National Science Foundation. And there are a number of instances of Symbiota. There is a version of this for the fungal community. There are versions of this for the insect world. And then there are versions of this for the vascular plant community. And so we have a consortium of herbaria, referred to as the Consortium of Midwest Herbaria, that are using an instance of Symbiota to house our digital resources. The features of Symbiota include a archive management set of tools that are kind of behind the scenes. I'm not going to share anything about those today, but also includes a number of public features, and that's what I want us to spend some time on. Within the Midwest Herbaria archive, we have a series of high-resolution images, so that each of the images each of the specimens in our IU collection is available online as a high-resolution digital image. We have a digital database that is fully searchable. Then there are tools that can stand alongside this database, including species pages that provide in-depth information about each of the species. We can create checklists that are broken down by a particular geographical region or a site that's of particular interest. And there's also an identification tool, which I want to show you near the end of my program today, called the Golden Key, which is extremely valuable for the general public using this web platform. Okay, well, how do we get to this point of having a digital archive on Symbiota? And starting in 2014, we launched a 5-year effort to modernize the IU Herbarium. It began with my first spending 3 years curating the collection, bringing everything up to date as far as the names that the plants bore, making sure identifications were correct, doing some rearranging of the herbarium, and basically making sure the collection was in excellent condition as we went through the process of digitizing it. Phase 2 was one involving actually imaging each of the specimens, and as we did that, to create a skeletal database. Phase 3 then was to flesh out that database using optical character recognition, OCR, to gather the label information, and then with student workers to edit and flesh out that database. Then phase 4, once we'd completed phase 1, 2, and 3, was to georeference each of the specimens to its latitude and longitude so that it could be easily visualized on maps. And interesting, we were to do this in 5 years. So a very major undertaking of something of this scale and complexity. So I liken this to Pilgrim's Progress. If you're familiar with John Bunyan's work from several centuries ago, there were times when we were stuck in a slough of despond. There certainly were times when we thought we would never get across the river of death. But believe it or not, in 5 years, we did reach the celestial city, and we did it on time and under budget by about $100. It was definitely a team effort, and it could not have been possible without a cadre of about 100 of our undergraduates here at IU, who devoted a lot of that labor and sweat in making this all possible. Well, next, what I'd like to do is show you a few of the aspects, a few of the features of this digital archive. And because once you see these, then you will more fully appreciate why we wanted to mount the photographic scavenger hunt. So there are 4 tools that I want to quickly show you from our database. The first one is the physical database itself, which is searchable. And it indicates here that it interlinks many US herbaria. So for example, here in the Midwest, the major herbaria in our area would include the Field Museum, the University of Michigan, the Morton Arboretum in the Chicago area, and the University of Wisconsin in Madison, as well as our own IU collection. So these 5 herbaria together amount to some number of 865,000 specimens within this searchable database. And they can be searched by, as you can see here, the species, both scientific name and common name, by locality at any resolution that you wish, all the way down to the latitude and longitude, by the collector, by date, and by whether it's a type specimen or not. So it's a very flexible, in-depth search function. The web platform then includes some of the typical things that you would expect from a traditional manual of botany. I show here Gray's Manual of Botany, which was published in 1890. This was actually the volume that Charles Deane had early in the 20th century to do his groundbreaking work on the flora of Indiana. So a typical herbarium of this type, or excuse me, manual of this type, would give you the scientific name, perhaps a couple of synonyms, some common names, and notes and descriptions about the plant. However, it would not give you any vision of what the plant looked like in terms of live pictures of live plants or herbarium specimens, nor mapping functions. So these were unique traits, unique aspects that we could supply as tools with our digital database. Plus, we can make it more colorful and more detailed than could fit into a single manual of that type. So let me show you a species page from our website. And I chose the tulip tree, which is Liriodendron tulipifera. I chose this because it's our state tree of Indiana. And there's a scientific name. There's a common name, tulip tree, but it's also known as tulip poplar and yellow poplar. So if one were to click on the More button here, you would find a list of other common names that have been applied to this particular species. It has a primary photo that shows the flower and leaves of the plant, and you can tell perhaps that it is a tree. There are secondary photos that show other aspects of the species. And as you would scroll down, you would see additional images that show herbarium specimens of tulip tree. In this area, there's text information, which provides a detailed technical description of the plant. But you could also click on other tabs, V plants or Gleason and Cronquist or Indiana flora. And here we have the notes that Charles Deane supplied in his 1940 flora of Indiana. His notes tended to be more ecological, more folksy, more ethnobotanical in nature. Finally, in the lower right-hand corner of the species page is a icon that shows a mapping function. And if we click on that, it opens up a new window that shows where the tulip tree, the tulip poplar, grows by querying the database that we've created, that in this case is using all of the herbaria that are part of this consortium. You might notice that that overall the distribution is in Eastern North America, but there are a few outlier dots. For example, one that's out here in Colorado. And if you were to click on that dot, it would open up the specimen record, and you would see that that is a cultivated specimen that's in the University of Colorado's Arboretum. If we zoom in on that map to just highlight see the Indiana region more close at hand, and we can see that most of the collections are in the southern part of Indiana. We could click on any one of those, as is done here. It would identify that that's one of our Indiana University collections. We could further then open up that collection, see the specimen, see the label, and enlarge that down to where you can even see the dust of pollen grains that are around the base of this flower of the tulip tree. So many things that are possible using the species pages that are within our digital archive. Beyond the database and the digital and the species pages, we can create checklists that show sites and regions of interest. And these are being used by some of our state agencies, by some of our land trusts, by our own university, and by people who are doing floristic studies in parts of Indiana. I would point out that these checklists can be public, or importantly, they can be private. So for example, we have the Indiana Rare Species List, which is kept private because those species are linked to the voucher specimens that show where these species have historically been found. And we do not wish to share that information too widely. We want people to protect that information. And then a fourth tool that's part of this Symbiota web platform is the golden key that's useful for plant identification. And this particular tool can draw from any of the plant lists that are already on the site or that are generated by a search of the database. And the plant key attempts to use simple language to make it appeal to the citizen scientist. And it functions by simply checking off traits that are in one column. And as those traits are clicked off, then the species list in the other column gradually gets reduced in number until the candidate species are few enough that hopefully you can know exactly which species you're trying to identify. Okay, now with that background, I can finally turn to why a plant photographic scavenger hunt. If you were to look at those species pages a year ago, we have about 2,700 vascular plant species in Indiana. But a year ago, only about half of those had color images that showed the details of those plants as they would look in the field as live plants. Most of the others, 50%, had just herbarium specimen images, which are fine for those of us in the technical areas, but certainly do not speak to the general public. Dead flat plants mounted on a sheet of paper just doesn't work for the general public. So we wanted to complete populating all of our species pages with colorful field photos that would really make these plants pop and be useful in the eyes of the public. So species pages was our primary goal. But also these same photographs would be useful in the Golden Key and other places in the website, including the glossary. So our objectives in the scavenger hunt, our goals, were first of all to obtain images that would be useful for the web platform. But we hoped that this would have other byproducts as well. Secondly, that it would encourage buy-in from the public. As individuals participated in developing the website and learning more about the website, that it would get more usage and word would spread around the state of this valuable tool that's available to the public at large. And thirdly, we hoped that this scavenger hunt would add further to our knowledge of the flora of Indiana, because as individuals would submit photographs, we asked them to include information about where they took that photograph, and even to supply the habitat and maybe even the latitude and longitude where that photograph was taken. So these bits of added information then could add to the archive of physical specimens in terms of what we know about species and their distribution and their ecological setting across the state of Indiana. Okay, so the process of working the scavenger hunt involved, first of all, creating a hunt website. I'll show you some of the details of that in a moment. As the hunt website was coming to completion, then we reached out to partners across the state who would help us promote the project and advertise it to get participants to be involved. So we reached out to land trusts. For example, our local land trust is the Sycamore Land Trust. Environmental groups such as the Nature Conservancy. Native plant groups, there's an Indiana Native Plant Society. State agencies and property managers, our state parks and nature preserves, and master gardeners across the state. Using the Hunt website and these partners, we then started to see an influx of photographs. And my first and foremost role was to quality check those photographs as they came in to make sure that they were properly identified and to see whether they had enough quality that we want to include them into our website. And from there, they would go on to editing, where they can be cropped and enhanced using Photoshop. And then the photographs, and along with that locality information, could be posted online. So let me show you the Hunt website that we created. And here you can see our banner. It's time for the 2021 Photographic Scavenger Hunt, and pull on your boots and grab your camera, etc. Along the navigation bar, it begins with the hunting lists. And in creating these hunting lists, we decided that it would be helpful to break them down both by the location within the state, as well as where we are in the growing season. So Indiana has 9 ecoregions, or 9 physiographic regions, which are by way of saying that the plants that you would expect to be growing up here in the northeastern part of the state, say around Fort Wayne, in the Northern Lakes and the Bluffton Till Plain area, are very different from the plants we would expect to be growing down here in the southwestern part of the state, around Evansville, which is in the southern bottomlands and the southern lowlands region of the state. And using our database, we were able to then customize checklists for each of these ecoregions in Indiana. But furthermore, using that same database, we could even customize them down to what month of the growing season, so that we could update month by month the plants that would be in flower in your local region that you should go look for and see if you could photograph and then submit to our website. The second function on our website was providing how to find species information. Ideally, we wanted the public to learn more about the tools that were available on our Midwest Herbarium website, or the Symbiota website. And so we have information about the 4 tools that I just briefly showed you: the species pages, how to search the database, how to work the checklists, and how to use the golden key. So if you didn't know about the species that were on the hunt list, you could go look at the species page. If you brought back a plant that you thought was the right species, you could try it on the golden key and see if it came out. The third item that we had on our website were tips on how to take good diagnostic photos. So for example, the photo that's provided is trying to say, look, you want to make sure you're capturing the diagnostic features, because you clearly show especially features of the flower. Maybe though behind it, notice there are the fruits that are shown here. Have additional photos that show the leaves of the plant and maybe even the habit of the plant. So tips on how to take good photos, and especially ones that highlighted the features that we need to see in order to make that plant pop. The third function of the website was the information that needed to be submitted. And so we were encouraging the members of the scavenger hunt to not only send us the photos, but also to provide information about the where that photo was taken. And that was so that the photo could be part of developing a photographic observation, as shown in this particular example, in which I took a photo of turtlehead. It's a lovely native plant across the state of Indiana that I took in Marion County, outside of Indianapolis. And so by providing this locality information, and the photograph was, of course, time-stamped, on the website, we indicate how you can use your smartphone to find latitude and longitude. And I included the habitat, an open fen. And this information then becomes a photographic observation. So now we can begin to include these photographic records to stand alongside of the voucher specimens, the physical specimens that we have in the herbarium, to expand our knowledge of the flora of Indiana. So once you have your photographs, once you have your collecting information, then we have a place where you can submit those files. Give us your name, your email. We provide information as to how to zip your files together into larger folders, and hit the submit button, and they pop into our inbox here on campus. Moving across the navigation bar, we also included a progress bar, progress tab. Here we wanted to feature some of the best of the photographs that we received in a particular month, and give some updates on the statistics as to how the scavenger hunt was progressing. Okay, well, how did this scavenger hunt turn out? And we were actually very pleased. We thought it was quite a successful endeavor, as measured by the fact that we received almost 9,000 specimens, photographs, excuse me, by the middle of December, when the 2021 effort reached its conclusion. Of those approximately 9,000 photographs, about 2/3 were checked for identification, and then were edited and uploaded as becoming a part of the photographic digital herbarium. In the process of adding those over 6,000 photographs, we were able to eliminate approximately 900 species from our hunt list. So we started out with about needing 1,400, and we think we're now down to about 500 that remain. I thought very telling was that we continue to have arrivals, or we had some that came so late in the year that we were not able to process them before the project concluded for 2021. So we have another 3,000 plus records that still need to be processed and uploaded. We did have a few issues in completing the scavenger hunt, as you might expect, in that we were engaging the broader public. Some of the rare plants were definitely not going to be found and are still the group that are most wanting in our needs list. As I already indicated, we had this late season influx that still needs to be processed. And there was a challenge in reaching some audiences. We had hoped to have an easier time of getting connected to some, say, the state parks and some of the naturalists in the state parks, which proved to be a greater challenge than we had anticipated. But that aside, we had, I think, a very successful year. And we have, in addition to having been able to populate those species pages, some added benefits that go beyond just those primary photos. Let me show you several of them. First, we were able to add to the glossary with illustrations. We were able to improve the golden key, but as you can even see from this gallery of images that I display here, this is from a checklist that I put together for Trevlack Bluff, which is a nature preserve in Brown County that is owned and managed by Sycamore Land Trust. If I had shown you this gallery from a year ago, probably half of those images would just be herbarium specimens. But now you can find whole swatches of that checklist, which can be illustrated with these nicely colored and beautifully imaged examples of what each of those species looks like. But I wanted to show you in particular how our golden key has been enhanced by these photographs. In that, if we take a look at a specimen that I observed at Trevlack Bluff on that particular property, the plant in question, as you can see, has white flowers. They're fairly small. The flowers are arranged in these umbrella-shaped clusters. And down at the bottom of the inflorescence, where all the rays of this umbrella come together, there are no leafy structures to be seen. It's an herbaceous plant, and it turns out on the right you can see a portion of the leaf of this particular species. As a botanist, I would say that leaf is a compound leaf. You can see that the leaflets are quite sharply toothed and have rather unusual veins that aim towards the sinus between the tips of the teeth, which is an unusual vein pattern. So what is this plant? And let's see if we can figure it out using the enhanced golden key. To start, on the left-hand side, on Trevlack Bluff, which is a checklist that we're using and where this plant was observed, there's a list of the species that are known from Trevlack Bluff, and originally the checklist has a total of 351 species. In the right-hand column, we have a list of traits that we can check the boxes that indicate what we know about the plant. And so if we just begin by checking the fact that it's an herbaceous plant, it's a dicot, the candidate species list is reduced in half, from 351 to 171 candidate species. Well, let's go ahead and check off another trait or two. If we check off that the leaves are compound and that the flowers are white, that total number of candidate species is now reduced to 40. Let's just do one more and observe that the individual flowers have a wheel shape. They're radially symmetrical. And that brings us down to 22 species on our list. And at this point, I want to engage the new magic. Let's go ahead and indicate, let's check the box to say we want to see the common names and we want to display the images. At that point, now we have a gallery of candidate species instead of just a list of names. And these can be rather fully populated because of the scavenger hunt. So there's 22 species. If we scroll down, we can see a number of them are in this family called the carrot family, the Apiaceae. But if we scroll down, we see further down things that are in other families that we can quickly eliminate. Surely our plant doesn't look anything like this tall thimbleweed or the white avens or even the spring avens. But let's click on one more trait, which is the number of stamens, which turns out to be 5. And at that point, we're down to 9 plants, and I can show you 6 in one image. And we can tell, well, our plant doesn't look like this. These bristly fruits do not fit the plant in question. It certainly doesn't look like this. Here's the inflorescence, and it has a sparse open look. Doesn't look like this, which has very different leaf structure, just 3 leaflets, and the teeth are rather small. And it doesn't look like this. There are 2 candidate species that seem to have some match with the one, the unknown plant that we have. This one, which has the white flowers that are in a broad umbrella, and it has these rather frilly leafy structures at its base. And then this one, which also has an umbrella-shaped cluster. From the image, we can't maybe tell that it does not have those leafy structures, but we can see the compound leaves that look more like what we have in our candidate. And indeed, that is the final answer. Now, if we weren't certain, we could click on the name Cicuta maculata and learn more about the species, or we could click on this name Daucus carota and learn more about that species. But it turns out the answer will be the spotted water hemlock, the most poisonous plant in the flora of Indiana. Well, to kind of wrap up the presentation on my part, the future of the scavenger hunt, we still need to complete the hunt list, and particularly looking for the rarer species. So here in 2022, that will be our focus. We want to try to reach out more to the state agencies, property managers, since particularly the state botanists and some of the naturalists in the state parks might be able to help us find a few of those rare species. And we hope that success will breed success in finding some additional funds so we can process those late arrivals and complete the photographic digital archive. With that, I'd like to acknowledge some important partners in this project, starting with the IU Institute for Advanced Studies, which was a primary funder of this work. Of course, the Indiana Academy of Science and the Indiana Native Plant Society also supported the project. And I'd particularly like to thank Eric Knox, the director of the herbarium, and the herbarium assistant, Laura White, for assisting me in the scavenger hunt. And so with that, my formal part comes to a conclusion, and I'd be glad to answer questions.
Thank you so much, Paul. This is fantastic. I've been fortunate enough to hear some updates as the hunt was progressing, but it was wonderful to see and hear more about this and to see the tools. I've used them a little bit myself and really enjoyed that. And I can tell that it's a really powerful tool and set of data that's available to all of us, even those of us who are not biologists, who are not specialists in botany.
Thank you. Yes, that's the beauty. It brings it to the world at large, and the digital archive and the physical archive now are broadly available.
Yeah, and so while we're waiting to see if any of our attendees have any questions that they can type in the chat or in the Q&A, I just wanted to say, kind of to build on what you just said, that one of the things that I love about this project is that it demonstrates the ways that community members beyond our local university, beyond the Department of Biology, here can engage with collections to aid both their own needs for knowledge. What is this plant on my property or that I came across when I was out hiking or visiting somewhere, and can also contribute to participate in strengthening the resources here for future research. And I think these digital tools just expand the possibilities in great ways. And I've had the great fortune to get to work in several different kinds of archives and to see what happens in terms of accessibility for people who aren't able to be here in person in working in collections, and the enhanced search options and the types of questions. I mean, that the golden key tool, being able to narrow it down with the series of guided questions and then to see visual guides is just phenomenal, and even to allow people to address additional types of research questions and expand partnerships with organizations. It's wonderful.
Yeah, thank you, Susan. Yeah, that's exactly what we feel.
Yeah. So I'm looking here, making sure that I don't miss any questions or comments. Okay, here we go. So we do have some. One of the questions is, will photographs of rare species help or hinder conservation efforts? That's a great question.
Yeah, well, I will comment first that the locality information, as I noted with the checklist, the locality information is kept protected. You have to have permission to access that part of the database. So I would have to suggest then that, I would hope that it would enhance, because people would recognize this is a rare plant and alert either the appropriate state agency of a rare plant that might be of interest for protection. This is a property that is needing to be protected. Or if it's a landowner that might say to them, I have something special and I want to be more involved with the conservation of this species. So I'm going to be optimistic and would hope that it add to the protection and benefit the conservation of our rare species in Indiana.
And do you want to also take a moment to sort of reassure people? I have heard people say, oh, I know where, I'm going to use a not completely rare but still valued example that I'm aware of, morel hunters in Indiana, the mushroom delicacy, and folks have to really know what they're looking for to make sure they're gathering the right thing. And I have heard with varying levels of seriousness, people say, I'm not telling you where my spot is. And I can imagine that with some of the plants, somebody might say, well, I know where that is, but I don't want to tell anybody. So what reassurances, you've said some of the things, the location information isn't shared, that sort of stuff. So if someone did know where that was and was debating whether or not they wanted to share that with the herbarium, what are some sort of reassurances that we can offer to them?
Well, as you noted, when we received the photographs, we asked for locality information, but if they wished to not share that, that was, of course, their decision. And we did receive some photos that were just good photos, and they had perhaps taken it some years earlier and didn't know exactly where it had occurred. And so they were fine. They added to our photo library, and the individual can make those decisions on their own.
Yeah, excellent. We have another question from one of our guests. It says, hello, can you share again how a community member accesses the database? Is there a link you could show us? So I'll work on getting a link, and if you want to kind of describe what they should be looking for.
Yeah, of course, if you just search IU Herbarium, you would find the herbarium website, and that would have a link there. But just to say it out loud, it's midwestherbaria.org. midwestherbaria.org takes you to that consortium of all the herbaria that's on that web platform.
Excellent.
That's it. Yeah, you got it.
Excellent. Thank you, Elizabeth. Elizabeth King is helping out, keeping all of our technological things running, helping with that. Thank you, Elizabeth. Another question is, now that you have images of the herbarium specimens, is it necessary to keep the original specimens?
Yeah, very good question, and that is a frequent one. And as I indicated earlier in the program, yeah, each of these is a genetically distinct individual. And there are features of the plant that you can only study having a physical specimen. We can actually magnify, say, pollen surfaces or epidermal surfaces. We can look at internal structure to some degree. And we can even, if it's a properly preserved specimen, we can extract DNA from the specimen. So the physical specimens still remain as exceedingly important for the scientific researcher, maybe not for the general public, but for the scientific research community. It remains an absolutely vital need. So photographs online, what that does is it helps even the research community, though, to focus their work. We will get individuals who want to borrow specimens who are at another recognized herbarium, and instead of having to borrow several hundred specimens, they may be able to quickly tell from our photographic library that these, say, 20 are the essential ones, and that really expedites their work. And then finally, they may be seeing duplicates, or there might be enough detail in the specimens that they see online that they can actually just send us information saying, no, that specimen should be called this, given my new research on that plant group. And so we're getting emails all the time from the research community.
And so kind of to build on that, are people in other states and at other institutions, other places doing similar work to this project?
Yeah, I would have to say that because we're a medium-sized herbarium, and we were able to mount a very concentrated effort over 5 years, that we are probably far ahead of many other institutions, and certainly the quality of what we've generated is quite outstanding. But the major herbaria are working on this, but they may have 1 million specimens or more, and so it's going to take them a while to get all this material digitized. But it's an ongoing effort, and has been for about the last 15 years or so, that the computer technology and the storage capacities kind of reached that critical point about 15 years ago.
And to sort of build on that, were there things in the process of this? I mean, as you said, 5 years is a very tight timeline, and the progress is really remarkable, and you are all to be commended. So I congratulate you on that. And I'm thrilled to see what all you have accomplished. But are there sort of lessons learned along the way, sort of things that worked really well that you're glad that you figured out, or things that now you have done those and you would say, okay, if somebody else were thinking about this, take this into consideration. You can save them a little bit of time.
Well, one of the things that worked well for us was the idea that we were using student labor. Now, you had to have a good workflow plan and good supervisory people. But if you laid out the workflow, the students could come in for a couple of hours, 3 hours, and accomplish quite a bit. If they were trying to put in a 40-hour week, that work would be so boring and laborious and mind-numbing that they wouldn't want to do it, or you might get a lot more errors in it, and so on. So by virtue of using that student labor pool, part-time coming in and just having a well-organized workflow, it worked beautifully. The other thing you should look at is the flow scheme that we used. The idea of curating the collection first. We noticed that some other people have not done that, and you run into some of the garbage in, garbage out syndrome, where you have plant materials that are inadequately identified, and it just makes a more messy database. So it should be curated first. But as you start the curation, you get their curation started and get maybe 3 or 4 months into it, and then you can start to have the photography following behind it. And as the photography goes on, you have overlapping phases. They don't have to go one after the other. We have overlapping phases, and that makes the whole operation more efficient.
That's great. And are you still recruiting student workers for the project? And how would students who might be interested get involved with this or find out more about whether or not it might be a good fit?
At this point, the digitization of the existing collection is complete. So where we are now is adding new materials, which, we had about 2,500 specimens that we wanted to add this year. And so that's a much smaller workload, but we need to have several students doing that kind of work each year. And so those specimens need to be mounted. People are submitting those specimens, need to give us digital records that can be easily uploaded into the database. So yes, there's an ongoing effort, but it's at a much smaller scale now.
Okay. And in addition to the photographs, does the herbarium want people to submit new herbarium specimens, physical specimens?
Yes, we'd be glad to. And some of the challenges, as with even the photographs, is training people in terms of what is a good specimen. And so we're delighted to have individuals do that. I would comment that one of the friends of the herbarium is a fellow who lives up near Martinsville, who is totally an amateur botanist, but we've worked with him long enough now that he's submitting some really excellent materials to the herbarium that are adding to the flora of Brown County State Park, where he has part-time function as a naturalist, his own county, Morgan County, and so on. So we're glad to work with the amateur botanists, but it'll take a little time to train them in what makes a good herbarium specimen.
Right. And if somebody was interested in finding out more about that, should they go to the herbarium website and look for contact info there and send a note?
Yes, you would contact the director of the herbarium, which would be Dr. Eric Knox, and he'll be glad to talk to you. Excellent.
That's great. And thinking about this type of a collection, a fairly specific type of collection, and we know that people can use this to identify the types of plants that they come across that are around their area and that sort of thing. What are some other types of research that can be used? I mean, I would imagine, is it possible, especially with a collection as long-lived as this, that's to be looking at things like how we're seeing climate change impacting our environment, and we're seeing different things show up in different places or no longer show up in places? Can we do that type of work?
Yeah, very definitely. There have been studies already that have dealt with the phenology of plants. In other words, when they come into flower, when they leaf out, and you can use historical records to trace how that timeline is shifting as the climate is warming. I believe about a month ago, though, you had work of Richard Hull, as one of our graduate students, presenting, and he is using historical records, especially by Charles Dean in the southwestern part of the state, to do he's doing a modern flora, fine grained flora, but it's going to be able to compare against the work that Charles Dean had done a century earlier and give us a sense of how that flora is changing, not only due to climate change, but other human activity. So yes, I'm really excited to see what he's going to discover, because you can go back to the Dean sites. Dean's locality information is excellent. You can go back to those sites and see whether many of those plants are still there today.
Yeah, and that was an outstanding presentation. And so I would encourage anyone who's watching this who might be interested in finding out more about that to look up that video as well, because that presentation was also recorded and made available. We were really glad to have Rich join us and share that information. And I have worked in the past in archives that deal with a lot of linguistic material. And interestingly, one of the things that sometimes gets collected when folks are out doing linguistic work, making recordings and documenting the spread of language and things, they might also be taking local samples of flora and fauna and doing that. And maybe they're asking folks in that area to tell them the local names for those kinds of things. So we see a lot of times that these types of specimens can also inform linguistic knowledge and other things of that type as well.
Yes, yes, indeed. And that's why we felt it was important to put Charles Dean's notes from his 1940 flora onto the website, because he often captured a little bit of that ethnobotany and other folksy information that I'm sure people broadly and within the research community would appreciate having accessible.
Yeah, so I think all of these things are connected, and it's just fantastic how many different areas of knowledge and research these collections can inform. And it's just a wonderful resource that we are so fortunate to have. And thank you and Dr. Knox and all of your other colleagues for the work that you've done to expand this and make it more accessible to those of us who are in the area, but not necessarily experts in this particular field ourselves. So so much for your time and sharing this with us. I'm not seeing any other questions. I'm stalling just a little bit here in case you slip in one last thing, but I want to make sure to extend our thanks to you. And it was very much our pleasure to partner with the Herbarium in supporting this great initiative. And we wish you continued success.
Thank you, Suzanne. And really appreciate the opportunity to be with you today. It was just a total pleasure on my part.
Excellent. Well, I'm not seeing any last minute questions or comments, so I will thank everyone else for being with us, and we hope that you will join us again for the future series that's coming up. And again, this recording will be available later. Please share it with others and let them know as well. So thank you very much from the IU Institute for Advanced Study. Take care, everyone.